So thank you for attend to this workshop. I hope you are enjoying all the amazing workshops that are running during those days. So today we'll be learning about the 3D segmentation in CellProfiler. I'm Paula Llanos. I'm a postdoctoral associate in Simini Lab in imaging platform at Broad Institute. The team that we are helping today in the activities in the last part of the session is Betsimini, Suganya Sivagurunathan, and Shatavisha Dasgupta. Okay. For today, the objective is to learn the basic of image processing in 3D, also segment cell compartments in 3D like nuclei, cytoplasm, and any other organoids. Also get measurements and features of our objects of interest, like the size, shape, location, intensity, distributions, and so on. Before to start, you should have installed CellProfiler. You can download from the website here, and also all the material you can find in this link that also I copy in the chat, and you can access the basically two folder. The first folder will be the material for the demonstration that I will show you, and the second folder with the name start with activity. It's for the second session. Then it's the exercise that you will work after the demonstration. So why use quantitative measurements as is very known? The human brain is not very good uh, making quantitative measurements or give quantitative information about what we saw. So, for example, this is a very basic example where if you check the nuclei here in the center of those cells, you see difference of brightness of the nuclei. However, because our brains is very complex and also relate the things depending on the context, we will see different brightness but actually are the same brightness. The same in the example that is below. If we observe the object C and D, we will see that it's different because the object also around that center object is different, but actually if we measure and we use quantitative measurement to check that site, it is exactly the same size. So that's why it's important to quantify. And there is many of features in the cell that can be quantified, of course, and we can check, sorry. Yeah. So in the fluorescence imaging, we can quantify the localization, then the distribution of the protein that we are studying, the protein level itself, the morphology of the cells. So we have 100 of other features that we can quantify. And for the same reason, to do something more automatically in this process is very important, because then you can get a robust system to quantify, so that don't change in any experiment. So you will have systematic pipeline to do that, and also you can do statistics and you can measure multiple properties at once. We can quantify also heterogeneity, distinguish subtle changes, even those undetectable by eyes. And also super important for us as a biologist, it is very faster and less tedious. So I hope you're already are familiar a little bit with CellProfiler. There is some previous amazing workshop that also introduced this software. So I will go a little faster for this, but I will just give you an idea about how this software looks like. This is the interface of CellProfiler. So we have a left panel here where we will drag the pipeline, and we will build our pipeline also from scratch. And the right panel here, you can find the setting for each modules. You can also here, depend on what module is selected, you can, if you select the image, you can also drag the image in that box. And when you select other modules in this panel, you will see the setting that you can change. If you have any question about the modules, we have this in the question mark. You will find a lot and very clear information about the parameters itself, and also help about how it works the modules. So then you can realize if it's how you can change the parameters, and that is written by biologists, so it's super clear and it's easy to understand for us. So this also is we can find here a button for start test mode, which is important to test your pipeline before to analyze all the image. So after you are happy with your pipeline, you can analyze all your set of image that you drag in your in CellProfiler. Also, CellProfiler aggregates all the modules in different categories. Sorry. So there is categories depending, I mean, the objects, the aims for that category. For example, for file processing, there is modules for image processing, object processing, measurements, data tools, and so on. So also, if you you can explore all the modules. Also, you can search directly the module that you are looking for if you know. So what happened when in general when we wanna segment in 2D in CellProfiler is very straightforward steps. So I will show you that to show the difference also between 2D segmentation and 3D segmentation in CellProfiler. So mainly the 2D segmentation in CellProfiler, you will apply you can identify the primary object, which usually is the nuclei, what is used also as a seed to relate that object with the rest of the component of the cells. So, and when you identify that primary object, you can also setting the parameters for the thresholding and everything just in one module. So it is very easy, I mean, easy to work with that. And once you are happy with your nuclei, then you can go for the next module, which identifies secondary object, which will allow you to identify the whole cell objects, including the nuclei. And those objects, the nuclei and also the cell, will be related and for those in the pipeline. And also, if you want to identify a tertiary object, for example, the cytoplasm, which is the cell shape and without the nuclei, like here, you can also work with that, and then it's super easy and straightforward. Then in the 3D is a little different. So we have some differences that we will point out during the presentation, and we will see also how it works with specifically in 3D images. So the first thing that we need to consider for 3D image is the kind of format or how you need to input your 3D image in CellProfiler. So as a general idea, you can load your stack as a .tif extension image. However, if you are analyzing multiple channels, it is recommended to load each channel separately in CellProfiler. There is other option also to load your 3D image that you can see in this link. And when you can find different way to load also your data specifically. And also there is information in the input module which you can explore. And also in the module help, you can find what kind of image are supported for 3D. Not all the modules in CellProfiler support 3D image. That also is important. So when you select one module, for example, identify primary object, you can see and you press the help for that module, you can see that what kind of image support. For example, for that module support 2D, however, doesn't support 3D. Then, for example, this median filter, which is very common, support 2D also and 3D. So be careful each time when you are working with the 3D image that be sure that that module supports the format that you are working on. So as I mentioned before, in CellProfiler we work with the image segmentation and object declumping in the same module, which is identify primary objects. That is in 2D. In 3D, we will do two steps. We use the threshold to segment our objects, and then we will apply other module, which is the watershed, to declump that object. So the workflow of 3D image segmentation, this is to get a very general idea because there is, as you will see in the demonstration, there is some step in between that also will depend how looks your image and that. But to reduce more the complexity, so I just want to point out that there is a first step for image processing, which will be very helpful uh, to work on our signal to make the object that we want to segment brighter and the rest dark. Then the second part, after we reduce the noise and we get proper image, we will go for the segmentation itself, that as I mentioned, we will segment the object and then we will apply a watershed, which will help us to identify individual objects, then declump the objects. And after we have that, we can get measurements of the objects and also the intensity inside of that object. So that is the idea to get the individual objects in the image. So for image processing modules, for the first step, basically any transformation that you can think of to achieve this is legal. However, this does not apply to image you're actively measuring, because the aim in the image processing is to get your object brighter, but to segment and get the object itself, but doesn't mean that we will measure that image. We will measure the raw image, your original image, right? Which is what we are interested to analyze. Then to achieve this, you may need to enhance signal that really is there and mask out bright signal that truly is. And the more transformation you do before the segmentation step, the more critical is to check your segmentation against your raw image to check that your segmentation makes sense with what you observe, what you check in your raw image. That is also what is something that we will see. So there is some modules that will allow you to do that, like overlay outlines, overlay objects, and that. So the image processing modules, and actually during the image processing, they work with the principle that my image should have my object of interest bright and everywhere else dark. That is, in this example, looks like very easy to decide what can be a background and what can be a foreground, and then what are my objects. This is the image very clean, right? So we can apply threshold directly and set an intensity to decide over that intensity will be my object and under that intensity will be my background. However, not all the images are like that. So some images are very noisy. So if we do that, if we just apply a threshold without any image processing, we can have something like that, where we cannot detect our nuclei separately. We just see the very big white part, which is the ROI. So the idea here is try to make our nuclei brighter, for example, and try to decrease here the signal coming from the cell to be part of the background in the end. There is many image processing modules that we can use to play a little bit. So of course, not all of them we are using like right now will depend on the image and the object that you want to segment. but you can explore there. And for example, we have modules to enhance edges or enhance or suppress features, which works on tubes and speckles and or dark holes, smooth, crop, and also on other advanced category, which also work with 3D. Again, you need to see if the model that you are checking works with 3D. So let's see to go to a pipeline for 3D segmentation. So that first pipeline that I will show now, you can get from the folder demo 3D monolayer segmentation. You can download that and also follow me meanwhile I'm showing the things. And if any questions, if you have any questions, please let me know. Um, this one. Okay. And you you can see CellProfiler, right? The window? No? Okay. I think, okay, I will stop to share and I will share again. Okay, no, this was not the pipeline that I was looking to show you. Okay, let's see. And now, is you see? Yeah, okay, so thank you. Okay, so in this image, we will, I already dragged the image here in the image. So you will find three images with those have three different channels. So if you double click here, also you can see the, sorry, I cannot see the whole window of CellProfiler because the zoom tab and hide that. So I think that is okay. Okay. So here, for example, is a membrane signal that you can see. There is some planes. Hmm? You still see the CellProfiler? Okay. Yeah, I think so. Okay. Sorry, I will try to do again. Um, now it's no, yeah, okay, finally. Sorry for that. Okay, so here you can see I just select the first image and you can see here the different planes. You can see also that the first plane doesn't have any information. For example, for membrane, we have here we have more membrane signal. And then also other plane without information. So just keep in mind on that. You can also explore for yourself when you load the data. And here, this is the for input, right, modules. So here you can define the metadata based on the name of the file. You can also load the metadata if you need. But if your name already have the metadata that you are interested in use, you can define in with regular expression that also if you go to the question mark, that will give you some idea to how to build that. But in this case, for example, um, the data that we are interested in is classify especially the channel for each image, right? To identify which channel is each image. So once you have that, you can also go here to names and types and define from the metadata and what is what channel corresponds to what, and you can assign the name for that. For example, the channel number 2 will be the DNA, the 1 will correspond to mito, and 0 will correspond to membrane. So also if you need to if you need to quantify separately in different groups the analysis, also you can define that. In our cases, we have just one group, one data set that I want to show. So first, I will start the test modes, and then you can be able to see each step. In CellProfiler, when you start the test mode, also you can open the eyes here beside of each modules, and also you can see the output. So I just opened the eyes for all the modules. So, but when you are building your pipeline, that is sometimes not necessary, right? But when you are exploring, you want to check if each step is working properly. Mm-hmm. That you create that. This is the second in the second activity, right? There is a one calibration image, so that you can create from Fiji or other software. And I'm not sure if you want to understand that, Maria, or not, but oh, yeah, that was about that. Yeah, because in the second activity, there is one file which is calibration, but we are not using actually, is to just relate the pixels with the micrometers. So just to change the measurement of the that we are obtaining. But okay, so in this case, the first step here, if we see, we will rescale the intensity. The original image have a very low intensity, as you can see. So when you rescale the intensity, that will be rescaled intensity to 0 to 1. Then we'll be able, with that we can check better also the intensities and the contrast. So you can see the nuclei like that. So in the first modules, what we are doing is creating the seed. First segment the nuclei itself also, and also create the seed, which will allow to relate the signal of the nuclei, the signal of the cells, and the component that we are quantifying. Okay, so after we rescale the intensity, we add a module of resize, which that will help us, especially in 3D, because the 3D images are very heavy. So if we work the 3D, this is the same, you will see just a little lower resolution, but that to segment our objects, it's enough good, because will allow us to go faster and analyze faster. So you can depend on the size and the amount of image that you have. You can do that, or you can just leave the size of the image as originally you have. So in this case, we decrease the X and Y size to the half. Then we need to, of course, remember to come back to the original size when we want to analyze after. So we will click here, resize. We here are applying this next modules. We are applying a median filter, which will allow us to blur a little bit the image. So what we need to do that is because the the nuclei here, as you can check, have a heterogeneous signal. So for if you remember the principle for the segmentation, what we want is to get our object brighter and the rest dark. So in this case, because we are not measurements, anything here, we just want to obtain our objects. What we need to do is try to get a more homogeneous signals inside of our objects, and also homogeneous signals in the background to can have a better contrast between your signal and your background. So we are using that. You can apply different median filter. This is just an example. Of course, with your image, you need to play a little more to set what is necessary, because maybe you don't have a very heterogeneous signal in your nucleus, and then you can just apply a smaller, decrease a little bit the blurry that you are applying here. So when we are happy with that, we can threshold in that. In when we apply a threshold, there is a lot of strategies. Again, if you use here the module help, then you can give some ideas about what each strategy do in the image and what can be better for your image. So in this case, the Otsu method was good. This is the setting that we are using here. So this is the output. So this is the median filter the output after the median filter. And in that image, we will apply the threshold. We will define the threshold to decide which is object and what is background. As I mentioned before, the if you have a heterogeneous signal inside of the nuclei, it can make some holes. Even if we apply median filter to avoid that, you can see here that still there is some holes here, for example. And we don't want that. We want more a solid object, right? Then to work with that, we will apply and add another modules with remove holes. If you are okay with just threshold, then just apply threshold and then you can move on to the next modules. Then also here, also as you can see, the objects that are detecting, there is, for example, these two objects, two nuclei, is looks like they are the same object, right? So we are interested here in identify individual objects. So to do that, to declamp the nuclei that we are already threshold. Yep. Yeah, and you can hear when you in the modules, in the input modules, in names and types, you can click here where it says process as 3D. Then you indicate there that is a 3D. Also, when you are adding modules and you click there that is a 3D, the CellProfiler also will give you a message that that module doesn't work because it's a 3D image, in case that the module just works in 2D, for example. Super. So, okay, so we are now trying to declamping our objects, right? And for that we can use watershed. That module will allow to declamping, and we select the previous image that we obtained after remove the holes, and we will see. And here, so we can see here now that we have individual objects. So now it's not just one, so not just the same objects, right? So we can detect also individual. We can see that how those nuclei are separated. So once we have already our nuclei separated, we will resize of our objects. If you remember in the second step, we resize to the half, right? Reduce the image to the half. So now we need to increase the image twice, right? To come back to the original size. And as I mentioned also, what we are interested in is, in this case, and to get the seeds of our objects. So the seeds in this image will be the nuclei, but also to get more seeds, then we can erode the objects to get a proper seed. So we will have our object and also we will have our seeds which come from our objects, okay? And that seed will allow us to relate the cells with the nuclei, and it's like the ID for the object. So if you wanna measure anything, then all of the measurement can be related to one cell, one nuclei, and what you need. Mm-hmm. Um, yeah, I mean, so for this case, as I mentioned before also, when we want to just segment the objects, even if we are losing some signal because we are losing the resolution, that for especially for the nuclei, doesn't matter very much because what we want is obtain the signal of the sorry obtain the shape of the object itself. So after that, when we are doing the measurements, we are doing the measurement in the raw image. So then the raw image doesn't lose anything, right? Mm-hmm. Yeah, also you can play a little bit with the parameters in the watershed itself also. So to improve that that declumping. And also if you are not very happy, you can also go even, I mean, to come back to the first step when you are applying the filters, also to try improve that. That's why also it's important. In general, this is this pipeline already work with for this image. But when you are building a pipeline, you need to come back and to the previous modules to improve and see what can improve better your result after. Hmm. But this is a great pipeline to start, for example, if you have something similar. Um, Thank you, Shata, for letting me know. And okay, so we wanna obtain the objects, the seeds, right? So to obtain the seeds, we will erode the object. So I will show you here. So this is the same nuclei, and you see, oh, we will lose a lot of information of the nuclei, but in this case, we want just relate we want just to have the seed to relate the nuclei with the cell shape in the end. So no worries about that. This will be just the information to relate objects. Um, okay. Once we do that, we will resize the image that we do before. Here we will convert the object to image because that will be necessary in the end to segment the cell. That will make more sense after. So I will just pass these modules. So now we are working, this is the first part of the pipeline, is to work more with the nuclei, right? To create a segmentation of the nuclei, create also the seeds, and now we are working with the cells, the cell signal. So here we wanna thresholding the membrane signal. This is the original image that we are giving as an input. And here we will, I just set this threshold method as a OTSU, 3 classes. When you have a lot difference between the intensity, not like the nuclei that is very clear the difference of intensity. When you have more smoothly difference between your background and the signal, OTSU 3 classes is very recommended because then the pixels that are in the middle intensity have you can decide if you wanna classify as a foreground or background. Okay, so we will see the result here. So now this is the original image, the membrane one. Here and here is the threshold for that membrane mark. But we are not interested actually in the membrane itself. We are interested in to get in segment the cells itself. So when you segment, the white is the object that the object that you are interested to measure, and the black is the background. So in this case, what we want is the opposite. So we will invert that image to have the opposite. Then here. So here we just invert the colors to get now we have the cells objects in white, which is what we want, and the membrane black. Then we will apply again remove holes. So that will clean a little bit some of the holes that we find in the cell, like just a small one like this, but not all the holes. You see that there is still very dirty, and as you see here, for example, in the first plane, you see that this plane and this part, the white area, is considering as an object. Is what is we don't want that, because there is not any signal that is interesting for us in these planes. So it's something also that we need to work on. And we will okay, sorry, I skipped one step. So we will remove some holes, and then here we will do some we will add the signal of the three channels that we have, the DNA, membrane, and mitochondria, to get the information about what is the zones in the area in each plane, and the planes that actually are the cells. So the planes in the area that there is not cells there, we want to avoid. So to do that, we need to create a mask where we'll discard the objects that are not part of the monolayer of cell that we are working. This is what we are doing now. So I already get the monolayer. I add the image. It is not very different, so I just want to give the information to that. We will decrease the size to that, because as I told you, this in the during the image processing to segment, those modules are very useful, especially in 3D, because now we are working with a very small amount of channels and not a lot of planes. But when you want to work with your data set, will be a batch with so many cells and planes that will be very heavy to process. So it's very recommended to use resize for these reasons. Again, this isn't, we are not resizing to measure any signal after. This is just to segment our objects. So once we resize, and then we will apply this module, which is closing. That looks like very strange, but actually will give you a lot of information about where the cells are residing in that stack. So for example, in this part, this image will show us that there is any like cell from here, and also from that zones and area and planes. So the idea here is not to obtain just that information, not obtain information about the object itself, just the space where the objects are in the monolayer. That will help us after to discard some objects that are just not necessary in our segmentation. So here again, after I closing, that is take a little of time. I mean, now was very fast again, but take a little of time. Then I will come back to resize, and then I will do a mask of that cloud that I show you. So I will see here. So we will threshold that cloud. Then thanks to that mask, we will obtain just the object, for example, coming from this area, and we will discard all the small objects coming from that area in black. So that is really helpful in 3D, because one of the problem in 3D is that a lot of signal that are coming from other planes on other area are just add noise to us, to our image. So we create our mask. Here, here we are eroding a little bit our membrane. to separate a little bit of the cells between each other. So we are eroding here, applying eroding, and then once we are okay, we can apply the watershed. The watershed will allow us to identify each cell, and then you can ask how the software can identify each cell separately. It's because in this watershed, in this module, you can include now your nucleases and the membrane image. So we are the mask that we create to discard the objects that are out of the monolayer. So here we are applying our seeds to relate the seeds with the cell and then also the nuclei. So you can use that and define that, and also up here I apply the mask will help us to discard the object, and then we can have here, as you see before, when we was segmenting the membrane previously, we have a lot signal coming from other plane that I mentioned doesn't help us, but as you see here, now we have just the cells that correspond actually to the cell area and anything else. So that gives you a more clean result, right? And now that we have everything, we have the cell object, we have nuclei, we have the individual object, right, and our image, then now we can go to what we are interested actually on is measure properties in those objects. So we can measure, for example, here in this, we can import measure object intensity. So with that modules, here you can select the image to measure. So again, it's super important that you select the image from your original signal, not the one that we use to process. So here I select the original image, DNA and mitochondria signal. And what I want to do with those images is measure the intensities in the cells that we segment and nuclei that we segment. So here you can see now. So in the image DNA, the object nuclei, this is each object that we segment in that image, then you will get a ton of parameters and features that you can get and quantify. Also for mitochondria. You can also, of course, to have another channel that you are interested on to measure, I don't know, membrane signal or other kind of signal, Golgi, just depend of your experiment, right? But once you have already segmented the cell and the nuclei, that it is easy just to add any channel. So there is a question?
Yeah. 3 channel Z-step could be incorporated as it is, or every channel of the Z-step needs to be updated or uploaded separately?
It's recommended to upload separately each channels in CellProfiler. So and it's not recommended to upload multi-channel, one CETA stack multi-channel in the software. Yeah, and there is also another options if you have, I think, movies and those you cannot see in the. Maybe after I will copy the link where you can find that information about how to input different 3D stacks. But yeah, the recommended one is separate the channels. And also there is a module to separate channels in CellProfiler also, so you can add that module there. So here we, for example, if we want to measure object size shape, here we can also measure not just the intensity inside of the objects, also we can measure the properties of the objects itself, like the shape of the cell, of the nuclei, the volume, the area, eccentricity, other features that give you information about the shape, if the circularity, right, the axis, and etc. And also super important is to see if our segmentation works okay. So, for example, here, this is a model to overlay. So the nuclei that we segment, we will overlay with our original image, and we can, this important checkpoint to see if the segmentation that we did correspond actually to the nuclei that we observed in the image, right? And that looks like it's very good. You can save also that image to just um, get right that information from your pipeline. And also here you can rescale the intensity of the membrane, because as I told you, this image have a very low intensity, so we want to rescale to 0 to 1. You can overlay also now the cell that we segment with the membrane signal.
Paula, there's also a
question.
So for selecting the cells that should be employed, the signal of the potential channels, where they need to select it?
The selection. Yeah, you can, and right, okay, yeah. In the input modules here, you can define because in this case, I'm not sure if that answers your question, but in the metadata, in the name of each file, here is indicated as a CHO, the number of the channel, and that will also, we can define what corresponds to each channel. So for example, I define that as a channel, and the channel number 2 I will define as a DNA. So you can define in the CellProfiler that information. I'm not sure if that responds to your answer.
I think we can move on. We can see if they have access also in the chat.
Super. Yeah. So okay. Then to save the image, before to save the nuclei that we segment and the cell that we segment, we need to convert the objects, because if the output that we get right after the segmentation, the objects, and then we need to convert to the image. Just remember, when you convert, if you want to save the object information and convert to the image, you need to choose the correct format, because if you choose, for example, binary, you will lose the information of the individual objects. So then after to convert, you can save the image there.
Someone's asking, how are you applying resized cell nuclear mass to original DNA image? Do you resize the mass of that to original image cells?
I resize during the pipeline. I resize all of the. You need to remember that, right? To always resize all the mask and object that you get to merge with your original image size. All the resize step that is in the pipeline, and also you can explore later. You can check that convert the image to the original size. So just we resize previously because it's less time when you compute the pipeline, but for everyone, just remember to resize after. And yes, then we can, as I mentioned, just save the image and then you can also export the information that we got with this module export to spreadsheet. This doesn't work with the test mode, but work when you analyze the whole batch. And okay, then now is your turn. So is you can see the slide, or is it still with the desktop? Yeah, okay. So So now the activity, you can find the activity that you will do for your own in groups. That is the name of the folder, activity 3D noise nuclei segmentation. So what you need to do here is just the challenge in this dataset is that the nuclei is a little noisy. So you need to play a little bit with the parameters to denoise properly and to can then segment that object and measures and get the measurements. So just follow the activities, follow the step in the PDF of the activity. So we are working in group. Each person will be assigned to a break room now. Then, and during the activity, it is very highly recommended that one of you share your screen with the group to discuss the steps. So me and my team, Shata, Beth, Suganya, we will be around also to the rooms to see if everything is okay. So yeah, I think we are ready to go to the break rooms. If you have any questions so far, you can let me know. And also we are be around the room, so you can also ask questions there. Okay. So just mention that if you need more help, there is a help button, as I mentioned, in CellProfiler. We have also some office hours in that you can also require, and we can help it to you specifically in the pipeline that you are building, and also you can use the image forums that also is not necessary just for CellProfiler. You can use that forum for any other question about any software that you are using for your cell for your image processing. Okay. Okay. No, so I think we are ready.